Let's keep going then. So then right here, we're going to continue now with the histology. The histology is the study of the tissues. We're going to have different types of tissues. As you can see here, obviously we have mucosa. The mucosa is just a membrane that produces mucus, right? Mucosa usually is simple squamous. We have that in our mouth, for example. Right here you have pseudostratified columnar, right there. And you can see here also the goblet cells right there. And right here you can see the cilia. In addition to that, we also have, as you can see here, stratified squamous. Right? You're going to have that in your throat, right? Basically, it says in here the oropharynx, this is basically your throat, right behind your mouth. Okay, so then we have here simple cuboidal, right? You have that in the bronchioles, for example. Simple squamous, right? We said that's in the alveoli. And you can clearly see here, right? Right there, right? That's only one layer of cells. Those are other layers that we're going to have in the respiratory system. These are layers, not cells. They're going to be submucosa and adventitia, as you can see right here. See some of the functions, pulmonary ventilation. So basically what this is trying to say is how the air goes inside your lungs and outside your lungs, right? And a different thing is gas exchange. That's how the oxygen gets into your blood vessels, into your blood basically, and how the carbon dioxide gets from your blood into the lungs. That's pulmonary gas exchange, right? Gas transport is going to be possible because of the presence of hemoglobin at the level of your red blood cells. And obviously, the gas exchange that we said is going to happen between the blood and the tissues. Okay, so then how does respiration happen? So this happens because of differences in pressure. This is the respiratory system, and we said the respiratory system is just a tube. So here's the tube that goes like this, and then it branches out like that. And this one goes up in here like this, and here's your nose. We have air out here with certain pressure, right? We have gases, and the gases provide pressure. So if you really think about it, guys, let's say the pressure outside is 9. If this is a tube, then the pressure in the lungs is going to be 9 as well. How come, therefore, the air goes in and out? Because the pressure right here in your lung is going to be, let's say, 3. How come this pressure drops to 3? Well, there are two options. Either you take the air from there, which there's no way for you to do it, or the other way to do that is that you increase the volume of your lungs. So what happens in our lungs is we cannot remove the air from there, but what we can do is we can make our lungs larger. So the diaphragm goes down, and when the diaphragm goes down, the volume in your lungs increases. Now this is larger. Since the lungs become larger, the pressure in the lungs go down, and now that the pressure on the outside is 9 and the pressure in the lung is 3, so that's how the air goes from the outside to the inside. It will go from high, 9, to 3, low, and that's how the air goes inside. Obviously, common sense tells you, how do you think the air goes out your lungs? Well, the lungs will go back to the original position. Your thorax will recoil back to the original position, diminishing the volume in your lungs. Therefore, the pressure increases, and that's what's going to push the air out. So people got some balloons and put them inside a little dome right here, and they call this the Bell's jar, right? So then the balloons are collapsed, but you see how they are pulling this down like that? And when they pull this thing like that, they see that the balloons actually inflate. When they let go of this membrane, then the membrane goes up, and you can clearly see how the air goes outside the lungs right there. Now, we have in here measuring pulmonary volumes and capacity. So, you see this volume right there? Tidal volume, right? Inspiratory reserve volume is when you take a deep breath. For example, the maximum that you can inhale is that one. The maximum that you can exhale is this one. Residual volume is the amount of air that remains always in your lungs, and you cannot exhale that. Okay, so then there is a device that we use in order to measure these volumes, and that's going to be called an spirometer. Well, right here, this is a handheld spirometer. The person obviously puts in here the mouth and blows the air in there. The needle moves and is going to indicate what was the volume. Those are the volumes. So in addition to that, we can also measure in our lungs capacities, right? Volumes and capacities are not the same. Let's say, for example, this is a bottle of tequila. What is the volume of this bottle? Let's say this is 2 liters. Let's say that this tequila is up to here. This is, let's say, 1.5 liters. Okay, if somebody asks you, what is the volume of tequila in this container? Then you're gonna say the volume is 1.5. But if somebody asks you what's the capacity of this bottle, you have to say 2, right? Because that's the total amount of volume that you can have in this container. Right? Capacity is the entire amount of volume that you can hold, and volume is the amount of air that you actually are gonna have in your lungs, for example. And here you have inspiratory capacity, functional capacity, vital capacity. You can see here some of what? Of the tidal volume, inspiratory reserve volume, and expiratory reserve volume. So and what is that? Approximately 3,500. Okay, but right here you can see the tidal volume, which is the amount of air that goes in and outside your lungs without you even thinking about it, right? What is this? This is approximately 500 milliliters right here. Now, obviously, we are going to have capacities, right? And in here you see how the arrows actually occupy 3 areas right here, 1, 2, and 3. Why? Because obviously we said that those are going to be the sum of 2 or more volumes, right? That's a capacity right there. How do we measure these volumes and all that stuff? We said with a speedometer, right? And the speedometer is the device that we're going to use in order to measure those volumes. That's an example of one of those devices right there. And that's that. Then we have right here pH and ventilation. Function of the lungs with the gas exchange. There's another very important function of the lungs. That is the control of pH. You're going to have to know this chemical reaction. The reason why you have to know this is because this happens in organs, in a lot of tissues that we have in our body. So how is it that the respiratory system controls the pH? Well, right here, what is pH? Well, pH is basically the number of hydrogens that you have in a particular solution. So right here, you have hydrogen. Then you have pH right there. So then what happens if you have too much hydrogen? Then your blood will become acidic, for example, right? If you increase the number of hydrogens. What happens if these levels of hydrogen that you have right there go down? Then your blood will become more basic. Let's say that your blood is becoming acidic. So then, fairly easy to understand is, if you block this hydrogen in here, if you do something whatever, if you sell the hydrogen, if you put the hydrogen in a cake or whatever, you get rid of that hydrogen right there, then you can definitely reduce the pH, therefore bringing the pH to normal levels. And that is exactly what your lungs are going to do. First, the hydrogen is going to be mixed with bicarbonate right there. This is going to give you carbonic acid. And then the carbonic acid will disassociate into water and carbon dioxide. Guess what your respiratory system is going to do with this carbon dioxide? It's going to exhale that. If you exhale this carbon dioxide, then you're getting rid of this hydrogen right there.